Hello everyone, welcome back. Today we're going to be talking about DNA, transcription, translation. So they call this the central dogma of biology. These processes that take us from DNA to RNA to protein. The reason this is the central dogma or the most important thing in biology is because all living things have DNA and they all do this process. They're all going to use DNA to make RNA, and they're all going to use RNA to make protein. This very complex and sophisticated and efficient process is conserved among all life forms on the planet. You'll see some variations of this process in things like viruses, where they might go from RNA to DNA, then back to RNA, and then to protein. But generally, it's going to be DNA to RNA to protein. So with DNA and RNA, because again, what happens is this first step from DNA to RNA, they call that transcription. Just like a scribe who might take an Old Testament Hebrew scroll and then make another copy of it in Hebrew, or someone might take the original works or early copies of Plato, and then make a copy of that. They're still putting it into the same language, so we call this transcription. RNA language is very similar to DNA language. There is just some subtle differences, but for the most part, it's going to be the same type of molecule, an information molecule. But then when we go from RNA to protein, we call this translation, because now we're going from this information to actual molecules that have a function in the cell. So for DNA and RNA, some differences we'll see here is that DNA is double-stranded. You'll see it has this double helix, this sugar phosphate backbone. It's going to have 5' and 3' ends. So that runs anti-parallel. One side runs one way, the other side runs the other way. DNA is going to have A, T, G's, and C's. And RNA is going to be a little bit different. It's going to have U, C, A's, and G's. So instead of a thymine, RNA has a uracil. The sugar's a little bit different. RNA is going to have a ribose sugar. DNA is going to have a deoxyribose sugar. RNA is going to be single-stranded. DNA is going to be double-stranded. And it's on the inside we actually see those nucleotides, or the nitrogen bases, sticking out. So protein, so DNA and RNA, they look very similar. There's some subtle differences, but for the most part, it's the same purpose and function of the molecules. Now, there are some RNA molecules that are more than just information, and we'll look at those. But when you get to protein, when you go through translation, proteins have these three-dimensional folds and shapes, and it's that shape or structure that determines their function. So it's very different. And these proteins get everything done in our bodies. You think about transport proteins, enzymes that catalyze reactions. They are all over the place, keeping us alive. Hemoglobin is a protein that carries oxygen through our bodies. Without that protein, we'd all be dead. So these proteins are keeping us alive as we speak. So DNA and RNA, they can both be in the nucleus. DNA in eukaryotes is protected in the nucleus, but RNA is just a cheap copy of DNA that can then be transported out of the nucleus for translation to actually take place. So DNA is more expensive, you could say. It's more high quality, more protected. RNA is a copy of DNA, so it uses a little bit cheaper materials, and it's more disposable, and it's single-stranded, and that's going to be used by ribosomes. Ribosomes are going to read mRNA sequences and build these polypeptides, which is another name for a protein. So if we zoom into the DNA, into that double helix, well, what does it actually look like? Well, the monomers of DNA are going to be nucleotides. And these nucleotides have a sugar, a phosphate, and a nitrogen base. So every nucleotide has those three parts. The nitrogen base part can change. We could have a G, a C, or an A or a T. It can be any one of those four options if we're talking about DNA. Now, the only caveat there is that if you have a guanine on that nucleotide, when it bonds to something, its complement, guanine or G is going to always bond to C. And I put 3 lines there to show you that it's always going to have 3 H bonds or hydrogen bonds. Now, when we go to another one, for example, you have A to T. A and T are always going to bond together unless there's some kind of mutation or problem. And that's going to have 2 H bonds holding it together. And you'll notice these sugar and phosphates on the nucleotides, they're all connected. So we call that the sugar phosphate backbone. So the backbone of DNA is sugars and phosphates. The center of DNA is all of these nitrogen bases being bonded together with those base pairing rules. 3 bonds for G to C, 2 bonds for A to T. And I always have people ask, well, can it be instead of T to A, can it be A to T? Yes, they can be swapped. It doesn't matter so long as they're across from each other. Another aspect of DNA is we say it's anti-parallel. If we look at this sugar phosphate backbone on the left strand, it ends with a phosphate on top, on that 5th carbon of the sugar. So we call that the 5' end. The end that's exposed, we call the 3' end, and vice versa. So on this side, it's exposed 3' end. Down here, phosphate sticking out on the 5' end. So we say that this DNA is anti-parallel. Parallel, but they're running in different directions. So here's a zoom in on one of those nucleotides. So if we tried to label what we see here, this would be a sugar, this would be our phosphate, and this would be our nitrogen base. We have some other names to describe these categorically. We have purines and pyrimidines. Purines have 2 rings, if you'll see here, in their molecular structure. Purines, another way to remember, well, pyrimidines, we'll look at those. Those have 1 ring. You'll notice that. And they also have a Y in them if we're talking about DNA. It's one way you can remember pyrimidines, except for uracil. But you know that uracil is the replacement for thymine in RNA, so you know they go together. So anyway, you have pyrimidines and purines. Purines always connect to pyrimidines. That's why we get those base pairing rules. So again, zooming back out, you'd have those nucleotides with the nitrogen bases sticking out in the center, and then you have this sugar phosphate backbone running around the sides. And they call this the ladder analogy because it looks like rings in a ladder that you could climb. And then again, we have those base pairing rules. So one of the things you'll have to be able to do is match up if they give you a DNA sequence. Whoops. Say that's the DNA sequence. You'd have to find its complementary strand if you're doing DNA replication. So A will give me a T, T to A, G to C, G to C, C to G, T to A to T, T to A, G to C, C to G. So those base pairing rules are always going to be what you follow. Now the reason, or one of the first person to figure this out was Erwin Chargaff. And what he did was he looked at how many of each nucleotide different organisms had. And he noticed an interesting pattern. So if we look at a sea urchin, the adenine was 32.8, thymine was 32.1. That's oddly similar. It's almost exactly the same. You look at the G and C, also almost exactly the same. And that was true for everything he looked at. So what he said was, it seems like for every A, you have a T, and for every G, there's a C. So they call that Chargaff's rule, that those concentrations of each nucleotide are complementary to each other. And all has to add up to 100%. So that was a major contributing factor to determining the actual structure of DNA. And then Rosalind Franklin and Maurice Wilkins with their Photo 51, which you see there in the center, where they used X-ray crystallography, and they got this spiral pattern that looks like you're looking at a double helix from the top, and it's spiraling down to the bottom, like a spiral staircase. So that helped Watson and Crick figure out that this was a double helix and it had a spiral where every A equals every T and every G equals every C. They originally did this with cardboard cutouts, and they'd move around on their desk to try to figure out the arrangement. Then they eventually had these aluminum pieces made when they were more sure of what the structure looks like. And for a while there, they had a real issue with figuring out if the nucleotides faced on the inside or if they faced to the outside. And that was like the big breakthrough when they realized they actually connect to each other on the inside rather than sticking out on the outside. But at first, everyone thought the the nitrogen bases, rather, would stick out on the outside. So again, there's just kind of summarizing everything you can see with a little bit more detail. You see the A to T's have 2 hydrogen bonds. The G to C's have 3. You'll notice they still have, this is 1 nitrogen base. And then you have a sugar and a phosphate. So you're seeing that pattern consistent throughout. And actually that phosphate goes with the nucleotide above it. This would be the phosphate that would go with that nucleotide. So anyway, we're going to practice some of these procedures. So what's going to happen is one of the processes that needs to occur is through mitosis or meiosis, all the DNA in S phase is going to have to be replicated. So we'll look at how that actually works. So for DNA replication, right, we've got our classic DNA double helix. Well, to replicate it, it needs to be opened up. So we have an enzyme called helicase that actually slams into those hydrogen bonds and separates the DNA. Now the DNA would click right back together, but thankfully there's something called, and I don't think it's shown in here, single-stranded binding protein that holds the DNA apart so it doesn't latch back together. Then you have all these other enzymes that come in and try to help facilitate synthesizing new DNA. And one of those is DNA, whoops, it's going to let me go back. Sorry. One of those is DNA polymerase. So DNA polymerase, to polymerize something is to make a polymer. So this is going to make a polymer of DNA. So it's going to come into those exposed DNA segment, that original template strand, and it's going to add these complementary nucleotides. So if it's A, T, G, C here, then it's going to add T, A, C, G as it builds a new strand. Now, if it's going in the direction of the replication fork, because DNA can only be made 5 to 3 prime. So if it's moving with the direction of the replication fork, because it can actually make it in that 5 prime direction, 5 to 3 prime, well then it's going to move in the direction of the replication fork. As this continues to open up, that will be able to continue to move because it's moving in the same direction. However, on the other side, it still has to be made 5 to 3 prime. So it's being made in the opposite direction of the replication fork as the DNA is being separated. So what happens is that DNA polymerase makes a short segment, and then it has to catch up to the new exposed DNA and make a short segment. And something I forgot to mention is that for DNA polymerase to even be able to access this DNA, a primer has to be laid down. So a primer is a small section of RNA that tells the DNA polymerase where to access and start synthesizing new DNA. So going back to summarize, we've got single-stranded binding protein, holds it apart, helicase, separates the DNA so that it comes apart, and then you have DNA polymerase and RNA primer, or primase. Primase adds the primers, so DNA polymerase can come in and add DNA nucleotides. And then the last thing that will happen is these Okazaki fragments that are made on that lagging strand will have to be sealed together with glue that is produced by ligase. It glues the little gaps. So this is what that looks like. So we've got helicase coming in, opening everything up. See if it's going to let me play this little video here. So helicase is opening it up. Then you've got primase added that primer, and then DNA polymerase 3 can come in and synthesize new DNA. And then DNA polymerase 1 will come in and replace those primers with DNA, because the primers are RNA, And then DNA ligase can come in and seal the gap. So going through it again, separate it. Primase adds a primer. DNA polymerase adds DNA. DNA polymerase 1 comes in and replaces the primers with DNA. And then what's not shown is ligase would come in and seal the gap. It actually is shown. So you'll notice on the top you have that leading strand that is going continuously with the separation at the replication fork. And then on the lagging strand, you have those Okazaki fragments being made that will then have to be sealed. This is what it looks like full speed. This is a little computer animation. Obviously, this wasn't really filmed anywhere, but it's showing you how fast it can actually go. They can add about 500 nucleotides per second to the growing replicated DNA strand. You'll notice the lagging strand is peeling off the top there. They're making it in segments. You'll see all the different enzymes coming in and operating here. And then on the bottom, you have that leading strand that's just made continuously. So it's made much faster. So they're adding 500 nucleotides per second. Super efficient, absolutely incredible. All of these different numerous machines that are automatic and just working and grinding to make this process happen as efficiently as possible. And it makes very few mistakes, although our bodies do have a mechanism for going in and fixing errors that occur. So all of that was DNA replication. So that everything we just looked at was going from DNA to DNA. So that's DNA replication. Now we're going to look at what's called transcription. This is when you take DNA and you build an RNA transcript. So you'll see here the DNA still has to be separated, but in this case you're building RNA instead of DNA. So it's the same process essentially, except that this little mRNA transcript is going to be much smaller and it's going to be released once it's made rather than forming a new double helix of DNA. So it's going to be single-stranded. The nucleotides that it will use will be A, U, G, and C. And that's one way you can tell if you have RNA. Does it have uracil in it? If it does, it's RNA. And here's a little animation of transcription happening. You'll notice now it's a single strand that's being produced. The nucleotides are being added. Now they're using, see, it's A to U, G will be C, C will go to G, so on and so forth. And this is slow motion, and now it's going to show you it at the actual speed that it would go, about 500 nucleotides per second. Just look at that thing burning out the nucleotides. It does it super efficiently with very few mistakes. And it has to be done perfectly, near perfectly, to keep our bodies alive. Very small mistakes, and they can have very catastrophic effects. So we'll practice making some RNA. So we got our DNA strand. So we'll come in here and add some RNA nucleotides. So remember our base pairing rules. DNA to DNA is going to be A to T, G to C. If we're making RNA, it's going to be A to U, G to C. So T will go to A will go to U, C will go to G. Now it's important to remember DNA is not becoming RNA, right? It's being used to make a copy of RNA, right? We're producing something else that's being released. The DNA is not being magically turned into RNA. So then C will go to G, G to C, C to G, T A, C A U, C A U, U U U, G G G, U A G. And that would be our mRNA strand. And then, so DNA to RNA, we just modeled transcription. And if we took that and tried to make protein, we would call that next step translation, going into a new language, going into protein language. Now, when that RNA transcript is made, thankfully, it can be modified in several different ways. So what happens is that gene has these segments called exons and introns. The exons are actually used to build the mRNA, and the introns are spliced out. So there's these post-transcription modifications. Everything's copied at first. We call this the pre-mRNA or an immature mRNA. Then the introns are cut out, and we splice together the exons. and then a 5' cap and a poly A tail are made so that the mRNA molecule can be recognized by a ribosome, and so it can also be protected from unraveling. So how do we go, when we get that mature mRNA, how do we actually go from that to protein? Well, we use these codon charts. Now, our cells don't need these. They don't have to compare it to a chart. It's just all automatic with these anticodons. But for our purposes, we use these codon charts to figure out what amino acid each codon or each set of 3 letters in the RNA will actually code for. So the way it works is you always start on a start codon. Your start codon is going to be AUG. So you're searching for those 3 letters. Once you find them, that's going to be your first amino acid. The way you read these charts is inside out. So if the first 3 letters are AUG, I would start in the middle of this codon wheel. I would find the A, I would find the U, then I would find the G. AUG codes for methionine. That's being abbreviated here as MET. The next one is AUC. So I'd find the A, find the U, find the C. Here it is right here. It's being abbreviated ILE. The next one is UCG. Find the U, CG, leucine, and let's see. Oh, I'm sorry. I did UUG. UCG, serine, and we would find it there. Then once you find a stop codon, UAA, then it stops. So that's not part of the protein. This would be the actual protein in the end. This is how it actually works. If we look at the ribosome involved, the ribosome has this small subunit and a large subunit. And it has these 3 sections, the E, P, and A sites. And those 3 sites bring in tRNA molecules that have this anticodon sequence that is complementary to the mRNA sequence so that they can stick to that mRNA molecule. A tRNA, that's a transfer RNA. They're made of RNA nucleotides, but their purpose, they look like the letter T, and their purpose is to connect to amino acids and bring in amino acids to form peptide bonds to build these massive proteins, or what we call a polypeptide, because they're made of many peptide bonds. So the ribosome literally just moves every 3 letters and brings in these tRNAs, and then the tRNAs exit. When the tRNAs come in, they bring amino acids. I'll show you this little video showing you what that looks like. tRNA molecules come in, anticodons complementary to the mRNA. Then on the back side, those amino acids forming those peptide bonds. You see that red to purple peptide bond, purple to blue peptide bond, so on and so forth. And when the tRNAs get into the E site, that's the exit site, they bounce off. When they're in the P and the A site, P is the peptide, that's where the peptide bond forms. A, I call that the access site. That's where the tRNA accesses the ribosome. So the ribosome is just sliding super quick, and the tRNA molecules pop out. So let's practice. AUG, AUG, methionine, MET, CCC, proline, PRO. Can barely read my handwriting here. UCG, serine, GGG.
I didn't get that.
Well, thought Siri thinks that serine is the same thing. Glycine, and then UAA is going to be stop. So that's actually not going to be part of the protein. This will be the protein. Now, how long are these proteins? At the very least, they're going to be 150 amino acids, usually significantly more than that. So again, that's what's happening there with those tRNA molecules coming in. Now, the DNA is not always going to be perfect. One of the things, mutagens can mutate or change the DNA sequence. So let's say this is your normal DNA sequence and you were exposed to some radiation. Well, you could have a substitution where a letter is replaced with a different type of letter. So it's supposed to have a G here, but maybe an A was substituted. So we call that a base pair substitution. That could, if it's in the first nucleotide, it's probably going to change it will change the amino acid that it codes for. If it's in the third nucleotide, it might not change it, so it could have no effect. But if it does have an effect, there's over a 99% chance that it's going to have a negative effect on the actual DNA or RNA. Here's an insertion. This is when a nucleotide is added. So a nucleotide was added there. Then it shifts all the nucleotides after that because they're read every 3 nucleotides. So now everything gets messed up. And then you could have a deletion, which also causes a frame shift mutation, where a nucleotide is just popped off. So then everything shifts back, and all the codons are now being read incorrectly. So deletions and insertions are going to have much bigger changes than base pair substitutions. So we'll practice again. So if this is our DNA, we'll make a complementary DNA sequence. So T to A to T, C to G. So we're making complementary DNA. So we're going to use DNA nucleotides, ACG, CGC, AAA, TGC, TAG. Given the strand of DNA, fill in the mRNA, amino acids, and anticodons. So the mRNA from that DNA strand, T to A to U now, because I have to use uracil, C to G, T to A, CG, CGC, AAA, UGC, UAG. The amino acids, we'd have to use the chart, so we can come back to that. But the tRNA, we would get from the DNA. So, I'm sorry, we'll get that from the mRNA, because it has to be complementary to the mRNA. So A to U, U to A, G to C, A to U, G, C, G, C, G, U, U, U. Let's see, that is a U, so we would have A, C, G, A, U, C. And then the amino acids, which I cannot see the chart from this page, so we will just have to load that in, see what we get. Methionine, and then the rest of these. So get the idea. Let me know if you have any questions. Thank you.